In this year's network, we have had a number of topics before us, but they sort of are connected. My talk is definitely connected with Peter Borger's talk as of yesterday, because Peter talked about the fact that there is information, even though it's very difficult to define it in biological things. There is information. And of course, then the question is, how did it come about in the first place? Where does information come from? Now, I don't want to talk about information today, because that was dealt with yesterday. And that has got the advantage that I don't have to use this difficult term as for precise definition. And you may have noticed already that I haven't called this talk The Origin of Life, but The Origin of First Cells, because I also want to stay away from the difficult definition of life. It seems to me that life and information are actually terms we can understand intuitively, but the moment we try to dissect them and define them in scientific terms, we don't no longer know how to use them properly. Intuitive knowledge is very important, but it doesn't always sit well with scientific enterprise. So we'll look at the origin of the first cells. In other words, the transition of dead matter to living matter. Some people also call that abiogenesis. How was living matter bios? Here you again have this term, living matter, life was generated. Just like this sand, you can see ripples in the sand and a jellyfish graciously swimming above it. The scientific disciplines in the question for the origin of first cells is chemistry and information theory, and I will concentrate on chemical aspects. Stephen Mann is a famous researcher in the field of origin of first cells, origin of life. I do not think that he is a theistic person. I do not know him personally, but from his papers, I would gather that he's a person that tries to approach this whole question from a purely naturalist standpoint of view. And this is a quote from a paper of his: "The study of biology offers no illumination on the origin of life." So this, again, gives us some kind of constraint, borderline. We need not look into biology. We are not dealing with cells that are able to replicate, that are able to mutate, that are able to adapt to their surroundings. We'll just look at how did the first cells come about. So we don't have to deal with biology in the sense proper. Again, in other words, we are asking for the origin of biomolecules and biomolecular networks. And we will also be looking a little bit into biochemical and cellular organization. You might call that hardware and software, but this is just metaphors that don't take us far. Just to illustrate, we want to know how did a primordial atmosphere organize into a primordial soup, so a collection of chemicals that was able to be converted into proteins, nucleic acids, and other chemicals that we know cells are constituted of. We know what we want to understand. The cells can be described. There are different bacterial cells, human cells, plant cells. They have a lot of differences, but we can sort of define a simple cell, let's say a bacterial cell, and know what it will need for the cell to have all its constituents in place. And obviously, we will not talk about how did the primordial atmosphere come about in the first place. So we won't ask further back, further to the left. There are a number of scenarios. People call that scenarios. Scenarios of how did it all begin. It didn't begin with a cell, so it began before a cell, and there are different scenarios. Some people would say at first in the origin of life or origin of first cells, first was replication, an RNA or a DNA, perhaps a protein. Some such chemical entity was formed, and it was able to self-replicate, copy itself. That was first. before all the other things also came into place. Some people would say, no, no, no. We at first have to discuss a metabolism, energy consumption and production. Small molecules, not replicating, but engaging in reaction circles. They're not forming chains that can replicate, but these molecules react with each other. So it's an assembly of molecules. And of course, we will need compartmentalization. All of this won't be stable if it's not contained somehow. That is what we call a cell ultimately. So some people would say compartments were first, liposomes formed, and inside the liposomes, which formed by chance and due to natural laws, inside these lipid proto-cells or pre-cells, the replication and the metabolism started. So this is 3 different scenarios. Most experiments and hypotheses that are presently purported on how first cells originated fit into one of 2 classes. Number 1, primordial soups. That is my subtitle. Soups. I did not invent this expression. It's quite common to talk about primordial soups. And people that talk about this in more technical terms would say polymeric molecules form first. And these polymeric molecules, not in the beginning, but sometime later, had the ability to replicate. This is what people call replicator-first or genetics-first scenarios. RNA world is another term where the replicator has already been proposed. And the second big scenario is the pizza scenario, where you don't have a soup, a reaction mixture, but more a surface where molecules can interact, and the surface kind of fixes the molecules so they don't swim away, they don't diffuse away, but can engage in reaction circles which can then develop further. This is called primordial pizzas by some people, and metabolism-first is a more technical term for it. I will now present these 2 scenarios in a rather cartoon-like fashion, so I won't show molecules at the moment. I will show a few molecules, but I don't want to burden you too much with chemistry. Some of us, including me, will regret this, but fact is that when I identify as a chemist, many people start by telling me when they stopped having chemistry in high school. I am very delighted to hear that they did not like chemistry, and I'm even more delighted that this is the first thing they want to tell me. Anyway, I will leave out the chemistry for a moment, and let's say this is the primordial soup, the mixture of building blocks. And by chance, they assemble to a chain of molecules, chain of small molecules forming a polymer. And for the sake of illustration, this is called start life. It's a polymer that forms from this soup by some chemical process, which we would need to discuss if it is plausible. This start life needs to develop the chemical property of being able to replicate, meaning we have more start life molecules in this mixture. If it doesn't replicate, it will be hydrolyzed or whatever the chemical process and just vanish. And then, of course, we also need to propose that at some stage start life mutated into something which, for the sake of illustration, again, I chose to call smart life. So smart life is smarter than start life. And that is supposed to be an advantage in the chemical survival of smart life over all the other polymers that might have formed in this soup. Through a process which people call selection, although it's not biological selection, it's just stability of molecules. Stability of molecules in a certain environment. It's not the whole process of selection as we find with organisms. Through a process of selection, start life vanishes, it's less stable or whatever. Smart life survives, but there is no life yet, so smart life just stays in the mixture. From here, and now I have to abbreviate what the theories, of course, explain in lots of detail, compartments have to form. So smart life has to be compartmentalized, otherwise the molecules will not be able to interact and ultimately form a protocell. A protocell, whatever it was like, there are theories but no experiments, whatever protocell was like, it must also have developed a metabolism. You see that here, BEDAC, we will turn to this in a moment. This is the metabolism that also needs to develop. So because a cell needs to have a metabolism, energy production, consumption of molecules and energy, all the things that happen in our cells. So as a summary, this is the replicator-first theory. And I modified this illustration after a paper by Robert Shapiro, the late Professor Shapiro. He was one of the most important researchers in the field of origin of first cells. He died a number of years ago. I took this illustration and modified it from Scientific American. Shapiro was not at all excited about replicator-first scenarios. As I said, I won't go into chemical detail now because I want to give an overview of the present status of the origin of first cells research. Now, Shapiro, definitely not from a theistic standpoint of view, said the spontaneous appearance of any such replicator, appearance of start life in a soup, the appearance of any such replicator without the assistance of a chemist, look at that, faces implausibilities that dwarf those involved in the preparation of a mere nucleotide soup. Because this start life and smart life is, if it's an RNA world, the letters stand for nucleotides, chemical entities which we call nucleotides. Nucleotides compose RNA. And many people think that RNA was first. So smart life was an RNA, short RNA, which developed into being able to replicate itself. But Shapiro said, even to prepare this nucleotide soup, not to have letters, but real chemical entities that can react in this soup, is nigh impossible. DNA, RNA, proteins, and other elaborate large molecules must be set aside as participants of the origin of life. Well, that's not a lot of detail. I'm telling you about it, just the conclusion. One of the most important researchers in this field came to have. And the same is true for a more recent review paper by Mukowska and co-authors. Quoting from their review, these sequential chance event scenarios cannot achieve kinetic coupling between subsystems and thus lack one of life's critical features. They are also unlikely to occur without human intervention. Notice, again, this person from a naturalistic standpoint of view notices that this is unlikely to occur, but he knows, or he has an idea of when it would not be unlikely if there was intervention. But they don't elaborate on this in the papers. I found that I should spend a few minutes on the famous Miller-Urey experiments when I do this talk, even though, as you will see, if you don't know already in a few minutes, this is no longer seriously discussed in origin of life, origin of first cells research. But it's still in high school textbooks and in many people's minds. This is Stanley Miller in front of the apparatus, which is schematically represented to the right. So it was an apparatus where he put simple small molecules, which were supposed to be present in a primordial Earth atmosphere. He put ammonia, carbon monoxide, methane, water, hydrogen, and they tried all sorts of mixtures, putting them in this apparatus and exposing this mixture to high energy, for example, electrical discharges. But there are hundreds of different experiments. This mixture underwent chemical reactions, and a cooler led to the collection of material down here, some material collected here, which turned out to be a mixture of water, of course, and organic products. So what did they find in these mixtures after hundreds and hundreds of experiments? The original experiment in 1953 was called production of amino acids under possible primitive Earth conditions. So did they observe the formation of amino acids? Well, they did. But what they got was a mixture of a lot of things. It was an oily brown mixture of many chemical compounds. There were more amino acid types than we find in proteins of living cells. Some important amino acids did not form. Altogether, these oily brown mixtures were comparable to a solvent waste container, chemical waste container, but they were not comparable. The composition of the mixture was not comparable at all with the molecular composition of living creatures. But the main problem with this whole thing is the following. The main problem is not that some amino acids formed, some didn't. That is, to me, not the main problem with this experiment. The main problem is, okay, so we've formed amino acids, but this is a blind alley if it can't go on. And what does it need for going on? It needs the polymerization of the amino acids to give peptides, proteins. However, the Miller mixtures, all Miller mixtures, including the ones that were produced in more recent years, contain monofunctional compounds that prevent chain formation. So now almost all people that are active in this field and people that read about it, like myself, would say the Miller experiments are a blind alley. The Miller mixtures don't bring us anywhere. I want to illustrate that quickly. For those of you that know about chemistry, this is, of course, not necessary, but I decided to keep that slide in my presentation. So if we have difunctional molecules with two functional groups that are symbolized by the protrusions or by the indentations, we can form chains. These molecules can form chains of indefinite length. Same is true if one molecule has two functional groups, and this, of course, symbolizes amino acids with an amino function and a carboxylic acid function, so they can polymerize to give peptides. However, if monofunctional molecules are present, as are in Miller experiments, they prevent the formation of long chains, because as soon as a monofunctional molecule is connected to a bifunctional molecule, the chain stops there. You can't form long chains. Miller himself, not very long before his death, said the following in an interview. Stanley Miller was interviewed by a person called Horgan, and Horgan, when he interviewed Miller, wrote, Miller remembered predicting shortly after he had done his first experiments in 1953, that within 25 years, scientists would know for sure how life originated. Well, the 25 years are over, he dryly commented, Miller commented. None of the current origin of life hypotheses seemed to convince him. He called them nonsense and chemical mind games. I'm afraid I have to agree. And that is also why many people, later than Miller's experiments, thought about metabolism-first scenarios. Replicator-first seems to not work. Let's try another first approach to the origin of first cells. And this approach is, of course, we also need some mixture of molecules, simple molecules, a primordial soup. And this soup, in this case, does not replicate, but the molecules are compartmentalized. They collect in small channels or on surfaces, and the molecules start forming, as I show here to the right, top right, the molecules form reaction circles. So the B and C are not connected to each other, but B reacts to give C. C reacts to give A. It's a metabolism. Like we have in living cells, we have that a lot. So some of these assemblies of molecules form metabolic circles that work, that are stable. And many of them, of course, don't form anything which is stable, so they just vanish. They are selected out. And then, of course, the metabolic circles, the ones that are more stable, have to be, again, put together with a replicator system, which in these scenarios is formed later. And then we have either the top or the bottom scenario. Top scenario with a white background, replicator first, and with a gray background, I have shown the metabolism-first scenario in this simple cartoon-like representation, illustration, as modified from Shapiro's Scientific American paper. What could such a metabolism be? In this case, I would like to show a little bit of chemistry. Many people say, okay, one of those metabolic circles might have been the reverse citric acid cycle. The citric acid cycle is very basic, very important for our cells. Without a citric acid cycle, most cells won't work. And it's very important for metabolism, for energy consumption and energy production. So for reasons that I don't want to discuss now, people say it was not the citric acid cycle which was very early in the origin of first cells, in the origin of the metabolism of first cells, but it was the reverse citric cycle. So the reverse mechanism that we now have in our cells. And they say that because inorganic carbon fixation is absolutely essential to sustain life on Earth. And this is what the reductive reverse tricarboxylic acid cycle can do. This is what it looks like when you look at the molecules. The citric acid cycle, which is, of course, connected to something outside, and it consists of a number of molecules and a number of conversions. So these molecules don't form chains like DNA, RNA, proteins. They react with each other. However, many people have pointed out, and I will now already start with critiquing this, and this will also show us some of the features of this citric acid cycle, whether reverse or forward. How can all these things concur? How can they come about at a certain time? And the sort of problem with chemical evolution, some people call that chemical evolution. The problem is the molecules can't wait for a long time. They will deteriorate, react in different ways. The molecules can't wait for 1 million years until the other molecules are also there. So there needs to be some concurrence, simultaneous occurrence of all the components. What about specificity? If you look closely, you will see that we have, for example, here a reaction that involves a double bond conversion, and here we also have a reaction that involves the double bond conversion. A double bond conversion or double bond formation from a secondary alcohol. The same is true here. Here it's a tertiary alcohol, and there is additional water elimination, depending on which direction you look at this. Now, this is two very similar reactions. So how can one of them occur, but the other not? How can there be catalysts like we have in our organism, in our cells, that will facilitate the way a catalyst does, the one but not the other? In our body, we have a specific enzyme for each of these steps. And if the enzymes were not specific, the whole thing would break together. Some of the most toxic compounds that have been invented are compounds that inhibit enzymes of the citric acid cycle. Because if we inhibit the citric acid cycle in our bodies, the whole metabolism breaks down. So how the specificity? What about parasitic, which is of course not a correct chemical word? What about side reactions that take out some of the molecules and convert them to something else? What about the genetic basis for the catalysts, which ultimately have to be enzymes, because we have enzymes in our body? So there are lots of problems. So people say it is completely unlikely that such a cycle formed before there were cells, before there was all the genetic material, the organization a cell represents. This could not form, not even simpler beginnings could not form by themselves. That's what many people say. And on this slide, I've collected some of the objections chemists have voiced against this. So again, this is purely scientific argumentation, nothing to do with bringing in God or a plan or a design, just purely looking at the chemistry. Metabolism-first scenarios are supposed to show pre-Darwinian evolution. They show major issues as follows. And let me just walk down the list with you without extrapolating and explaining each of them. No practical chemical substrate is known. The citric acid cycle is just, well, people think this might be the beginning of a beginning. Experimental data supporting the main hypothesis, the spontaneous formation of such reaction cycles, are lacking or very preliminary. The source of energy is questionable or not specified. It needs energy input for such a circle to operate. But how? Where should the energy come from? And if it's too much energy, it will destroy the circle. You can't just put in as much energy as you have, like as lightnings or very high temperatures that would destroy the circle. Chiral question is not addressed, and I will not address it now. Multiplication abilities are not truly present or unknown. How can such a cycle multiply? If there is one cycle, how should it multiply, replicate, sort of, although it's not replication in the sense we talk about it in the RNA world. Heredity is not clearly demonstrated or absent. How could a circle, such a circle, lead to other circles be formed patterned by it? A plausible site on early Earth is unknown or not specified. Where should this have happened? Without the cycle being disintegrated or destroyed, the system is unstable. The evolutionary path seems either complex or questionable. This is from a review paper, and there are lots of things in detail people have put forward to sort of uphold this metabolism-first scenario, but they all lack one or more of the things that are needed for such a circle to form and be chemically plausible. So I will leave this now, the replicator-first and metabolism-first scenarios, and just tell you there are other scenarios. Yes, there are other scenarios, but I will then focus on just one very recent thinking in the question of the origin of first cells. RNA world scenarios, amyloids, belong, of course, to the replicator-first theories. So if a person says, but the RNA world scenario, you can go back to what I tried to explain, although very briefly and just in a very illustrative way, about replicator-first theories. RNA world is an example of replicator-first. Panspermia, extraterrestrial organic molecules, to me just removes the problem to another place. But the chemical question, the chemical problems and challenges are the same, unless we say that on a distant star or a distant planet, chemistry and physics were different. But that is not what anybody believes, including Christians. Some people talk about lipid vesicles, micelles, that form first. Some just do computational simulations that work very nicely. That reminds me of what we do in drug research sometimes. We build very nice models in computer of proteins. And we then build models of such proteins targeted by potential inhibitors or potential modulators. And the models can be really, really nice. They work nicely. But when you do the experiment, they are not worth anything, because you always need to have an experiment to connect to the real world. So simulations sometimes are just fog. I already mentioned that time is a problem. I've had people say to me, well, this had a lot of time. So I would say, but I'm not asking for time, because time could be a problem with the chemicals. I'm asking for a mechanism. I concede any time you want. Show me how it could have come about. I won't talk about the origin of homochirality, and as I said right at the beginning, I don't want to talk about the origin of the genetic and other biochemical codes, because this is definitely something connected to information, and we heard a talk yesterday about that. So the origin of life, origin of first cells, no matter how you call it, faces a lot of problems. You see a lot of question marks on this slide. And wherever there is a question mark, we don't know how this could have formed, could have happened by itself. Wherever there's a question mark, we don't know how could this have happened by itself, purely naturalistic, without anybody interfering, with no plan, purpose, or intention involved. And you see there's lots and lots of question marks, lots and lots of question marks, which of course is very good for science. I'm not saying that the question marks are what leads me to think that there is God, or no matter how you call it, involved. Question marks are just interesting from a scientific point of view. We like question marks. We like problems. We like to investigate. So that is what people do and did. And you see, a lot of the question marks have been turned into exclamation marks. Wherever there is an exclamation mark, we know how we can do this. If you give me enough time and money, we can prepare a protein for you. Even the protein you want to have, not just any protein. We can do that. But of course, it needs some planning. We need to have a plan. We use simple example, amino acid 1, which has been protected and activated, connected to amino acid 2. Then we remove the protective groups and purify that a bit, and then add the third amino acid. So we have a plan, and we know where we want to go. We know what we want to synthesize, and we have a plan how to do it. So all the exclamation marks mean we know how to do this. We know how it could have happened, how we can do it. And actually all the exclamation marks, everything is not unknowns. We know through scientific experimentation how we can put together such proteins and stuff. Science thus leads to circumstantial evidence for the necessity of creativity, for the assembly of living matter. Because this is what we found out. It can happen if we do it, so to speak. Can we also prepare a minimal first cell? A cell with just the minimal number of components. Several hundred actually. Craig Venter and others have thought about and done experiments about how many components does a cell actually need? How many genes does it need? A few hundred. That's the minimal cell. Do you think we can prepare such a first cell from scratch, from inanimate molecules? I think it is possible. Why not? Humans have been able to build a lot of things. Perhaps we can. And this is what people try. People try this top-down approach. Bottom-up, that was what we've discussed so far. Replicator-first, metabolism-first scenarios, where we try to build a cell from basic chemical building blocks, bottom-up. But now we think about it, top-down. It's the alternative approach by reducing a simple cell, because we have a lot of cells at our disposal, to find its essential parts and try to rebuild it on this basis and understand its origin. Like Richard Feynman, Nobel laureate in physics, famously wrote on his blackboard in his office. The blackboard, just after he died, was photographed, and you can find this photograph on the net. And one of the things that was on Richard Feynman's blackboard was, "I only understand what I can create." He was an atheist and a physicist. But I think this also applies to what people try to do here. There are research programs like Max Syn Bio, which is a German research project trying to put together a minimal cell. And people that are purely naturalistically thinking about this would say we need a radical rethink. This is actually a quote. The headline, "A radical rethink is necessary," is a quote from a paper Leroy Cronin wrote for Science in 2016. In 2023, Cronin and others published a paper, and I would like to talk about this for a moment, of Cronin explaining his thinking when he tries to solve this riddle of how did the first cells come about. Now I have to read this relatively long extract from Cronin's famous paper, Assembly Theory. And it says, scientists have grappled with reconciling biological evolution with the immutable laws of the universe defined by physics. Well, when you read this sentence, you find that there is a number of worldview propositions. Universe with a capital U, or the other words don't have a capital U, not even physics, Alexander, has got a capital P. And immutable laws, that is, of course, also something we can't prove. I think it's true, but we can't prove that. So there are propositions. These laws underpin life's origin, evolution, and the development of human culture and technology. So it's a grand theory of evolution, not just biology, everything, including human culture. Yet they do not predict the emergence of these phenomena. Emergence, as in creation, we would perhaps use the word creation, coming about, emergence, that's his word. Evolutionary theory explains why some things exist and others do not through the lens of selection. That's a very short representation of biological evolutionary theory. To comprehend how diverse open-ended forms can emerge from physics without an inherent design blueprint, a new approach to understanding and quantifying selection is necessary. So one thing for us to take away from this is, and please share this if you can in your churches, with young people, with anybody around you, if the matter happens to come up, we do not know how first cells were formed. Because in the media, fairly often they suggest that we've almost already understood. I did a talk like this to a lay audience not very long ago, and before I did that talk, I watched 2 documentaries on German TV to see, okay, how is it presented to people? How is this topic presented to people? And the main thrust of both documentaries was completely wrong, because they suggested that we know, and this is not true. Leroy Cronin is one example. Shapiro, I've mentioned a few other people. The scientists that are working in this field don't say this, unless sometimes when they're talking to journalists, when they're broadcasting their views. And that is not genuine. That is not good. Assembly theory. That is, let's say, the latest, although hotly debated and refuted by others in the field. Assembly theory is this 2023 paper by Cronin and others. Nature 2023. Well, when it was in Nature, it must be true, mustn't it? But that is not necessarily the case. What they did is that they analyzed molecules by formalizing what a molecule is, so stepping away from the chemistry, so to speak, and they analyzed how many steps will it need to assemble this construction, happens to be a molecule. How many steps does it need? They conceptualized objects not as point particles, but they thought about formation histories. How many steps would it take to assemble any object, including molecules? That was their idea. And the idea was, if it has a certain number of steps, like only 15 or less, it must have an abiotic origin. A biological origin, sorry, I used the wrong word. It must have had a biological origin. If it's bigger, it can only be of abiotic origin. So that's the idea. And please notice they don't explain how things could have come about. They just present a method to categorize molecules and objects into biological, non-biological origin. So it's not exactly what we're asking for. And many people, including famous Stephen Benner, critique this heavily. Stephen Benner is one of the most important people in the origin of life research, has been for the last decades. I actually met him 2 years ago at the conference in Israel. The conference was called Potential and Limitations of Evolutionary Processes, and Stephen Benner gave the main talk on the question of the origin of first circles. He's a purely naturalist researcher. I don't know about his personal faith. He's not a Christian, he said that. But he's definitely not a non-naturalist researcher in this field. And he's really famous. And he said that this is completely false. His critique, which you can read here, is fairly long. And he completely dismisses this Nature paper for a number of reasons. And without going into detail, one of the things he said was that it is false that non-living systems cannot contain molecules that are complex. Actually, he says that in a conversation with Lee Cronin. Lee Cronin asked Benner, what is the biggest molecule that was ever found? And Benner said the Cullinan diamond. It's one of the crown jewel diamonds because it's one carbon molecule, you might say. And it's non-repetitive if you look in detail. It's just carbon, and it's huge. And Benner writes that Cronin was not satisfied with this answer because he was hoping for something which is nearer to origin of life things. Anyway, I think that this assembly theory doesn't really help us because the formalization of molecules and their formation, how can it help us to understand the formation of molecules? We just formalize these things. And there's more critique at its lowest level, and curiously, basic building blocks seem to be bonds, not atoms, Benner wrote. Basic building blocks seem to be bonds, not atoms. And the assembly theory speaks of assembly parts as joining operations, not of real molecules. So I think it's just a distraction. It seems to me that this assembly theory distracts from the fact that we don't have the chemistry in place, which we would need to understand how these molecules could have formed, whether it be replicators or metabolism circles or whatever. The chemistry is not in place, so people formalize this, turn it into a simulation, I would say, and then say, okay, this is how it came about. At this conference, I mentioned this conference in Israel 2 years ago, there were more talks of the kind, and they were dismissed by people that were there by saying, well, okay, this is interesting simulations, interesting philosophical considerations, but what about the real chemistry? Because in the end, some real chemistry must have happened. We can't get away from this unless we consider everything we have around us an illusion. I was reminded of something which I read 50 years ago. I was about 16 at the time. And here's the translation of what I remembered. It's a quote from Enlightenment philosopher, German philosopher Lessing. I don't know if people that are not from Germany have heard the name. Gotthold Ephraim Lessing. He's supposed to be one of the most important Enlightenment philosophers. He was not a scientist, just as most Enlightenment philosophers were not scientists. They came in between big scientific developments. Many developments, scientific developments, happened before Enlightenment. Newton, Kepler, Boyle, and many others happened afterwards. Darwin, for example. So this is a quote from Lessing's essay on the pursuit of truth. It's a bit complicated. Let's try. It is not the truth that any man is in possession of, or thinks he is in possession of, but rather the sincere effort he has made to get to the truth of it that constitutes a man's value. For it is not through the possession of it, but through the investigation of the truth that his powers expand, which alone constitutes his ever-growing perfection. Possession makes me calm, sluggish, proud. If God had all the truth in his right hand and the ever-active drive for truth in his left, although with the addition always and to err forever, and said to me, choose, I would humbly fall into his left hand and say, Father, give. The pure truth is only for you alone. So I think this is a very important psychological consideration we have to make when we present scientific apologetics. People will consider the quest for truth higher than knowing the truth. Of course, everybody likes to get at truth and get a nice paper out or some product really nicely produced. But scientists delight in the quest for truth, not in being told the truth. So when we present this topic, for example, origin of first cells topic, I would like to make it clear that although I do not do research in this field, I'm really interested in, as a Christian, how did God do it? Can we find out by investigating nature how this was put together? No matter how long it took or whether it took very short time. I'm interested in how is this built and how could it have come about, and can we perhaps build a cell? I find that very exciting. I know that some Christians are scared if I say that perhaps humans can build a cell, small bacterial cell, and it could have consequences, but a lot of technology has consequences. And we should perhaps, when we do scientific apologetics, always stay with supporting science. And I think this was at least part of what the speaker last night, Sharon Dirks, what she said, that we are not against science, but we're against some of the if people put science absolute, if they say only what we can find out in the lab is matters. And this is how I want to end my talk. Is it mind for mechanism or mind before matter? What do I mean by that? I think we have found out, and we are finding out more and more, that the origin of life and first cells was not something just causal and mechanistic, though it was accompanied by a mechanism. There must have been some mechanism. I don't know how God did it. Perhaps we'll never find out. Just as people that do not believe in God would say we'll probably never find out how it happened in the first beginning, in their worldview. They would admit to that. Even if we can do it, if we can have cells produced on their own by a purely naturalistic means, we still don't know if this is how it happened some billion years ago. But there was a mechanism, but this is not all that is needed. Research has shown that it also needs organization. Note the term self-organization, which comes up in almost all discussions on the origin of first cells. Some people call it integrative management. You can also call it construction of a cell. It needs attention to detail. It needs, whatever that exactly may mean, creativity. So acknowledging the evidence, not the gaps of our knowledge. The gaps are also there. Perhaps we can close some of them. Acknowledging the evidence opens up two choices. This creativity, self-organization, whatever you name it, is obvious. It is obvious and observable both by intuitive and scientific examination of the cosmos and its details. It's not a gap of our understanding, but an empirical and logical observation. And the more we come to know about living organisms, the more we understand that this is what is needed. We can't do this research and ignore that all the time we see it needs a plan, it needs an intention. We can't blind our eyes to this evidential fact that this is what is needed, at least according to our present-day knowledge. So who do we think capable of having original creativity? Impersonal, purposeless matter, or a personal, purposeful God? Certainly purpose. But if we want to know more about this God, we need to turn away from science. We need not ignore science, but now we need to look for other sources of knowledge. We can't find whoever God is and whatever he intended, we can't find that in science. We just find there is intention, there is purpose. Of course, this is all a corroboration of a Christian premise. It doesn't prove God, but it corroborates a premise, which I, as a Christian, have had all the time. And I'm glad to admit to that, if you haven't noticed already. Because it says in the Bible, in one place, that which is known about God is evident within them. For God made it evident to them, evident. For since the creation of the world, his invisible attributes, his eternal power and divine nature, have been clearly seen, being understood through what has been made. And in the Gospel of John, famously begins, in the beginning was the Word, and the Word was with God, and the Word was God. All things came into being through him, and the Word became flesh and dwelt among us.